and I am truly delighted to have you all here today. Thank you for taking out the time to join this live masterclass on primer designing and PCR optimization. I am going to share you the screen, then we start this master. Can you see in the screen? Can you tell me, do you see the screen?
Yes.
Okay, thank you. Okay, I'm going to share you the slides with you. So today's masterclass is about primer designing and PCR optimization. Okay, it is about how to design primers and how we can optimize PCR to get the better result. Okay, so it is from concept to optimization, a complete guide, hands-on guide. Okay, so first of all, I am going to tell you about me. My name is Daraz. I am doing my BS Genetics and Molecular Biology from University of Health Sciences. Brings a wealth of knowledge and hands-on experience. Bioinformatics, stem cell therapy, genomics, genetics, and evolution, molecular diagnostics, molecular biology, and so on. With a keen interest in advancing the scientific discovery, Ali is dedicated to inspiring others through engaging learning experiences and fostering a deep understanding of cutting-edge molecular biology techniques. Okay, what is the aim of workshop? Okay, it's actually unlock the precision, accuracy, and confidence in molecular biology. Okay, it gives three basic. It has three basic aims. First of all, the comprehensive understanding. Okay, you can get a complete workflow of primer design from selecting the target sequences to generating a high quality primer. Hands-on knowledge. You get to understand the PCR fundamentals, optimization strategies, gradient PCR, what is gradient PCR, touchdown PCR, the troubleshooting with the real world example. Skill enhancement. Equip yourself with the practical skills to design primers, optimize PCR reactions, and solve the amplification problems in independent. So let's start today's workshop. Okay, on this slide we have the modules of the workshop. First of all, we get through the PCR basics. Then we actually talk about the hybrid design, and at the end, none the other PCR optimization. Okay, first of all, start from PCR basics. So, what is PCR? PCR is actually the polymerase chain reaction. In the word, the polymerase means there are the polymers, okay, and the ase is actually the enzyme, okay, in which An enzyme is used to add sugars like the nucleotides, like the nucleotides to expand or to extend or to make replications or the copies of your DNA or the cDNA. Okay, and the word chain reaction is actually the cycle that is repeated 30 to 40 cycles like. So PCR is actually a selective amplification of a specific DNA sequence. Okay, we design like this is the DNA we have. Okay, but we have to just amplify this area. Okay, then we design primers. Okay, that attach to the specific area to amplify just this. Okay, so PCR is used to amplify the specific DNA sequence. So what is the PCR process? Okay, the one cycle is first of all we denature our DNA. Okay, so this is the double stranded DNA. First of all, we make it single strand. Okay, single strand, and then annealing. Okay, at annealing temperature, that is actually the primer optimization or the PCR optimization is actually we do to check where is actually annealing occurs. Okay, the best annealing. Okay, the primers attached at annealing temperature and then extension like the 72 degrees centigrade at which the maximum extension of your primer, sorry, the extension of the DNA strand, like from in 2n, like the exponentially increasing your DNA strands, like the one strand and there is a two double strand. So what are the requirements of the PCR? First of all, the DNA template that require having the target region. Okay, your DNA template must have the target region. Okay, like that. This is the DNA template, and it has the specific DNA target region. Sorry, and the two primers that actually attach with your DNA or the target. Okay, so the two primers are actually needed: the forward primer and the reverse primer. And a DNA polymerase such as Taq polymerase. Why we use Taq polymerase? Because they can bear deep high temperatures. And then we need TNTPs that actually attach with each other to extend the DNA strand. The buffer structure is needed to maintain it actually maintain pH. It actually maintain the pH of the solution. Divalent cations are also used to keep keep unbalanced the charges. The volume we use the 20 to 150 microliter in small reaction tubes. Okay, we need small tubes like the PCR tube. Okay, in which 0.2 ml to 0.2 to 0.5 ml volume placed in a thermal cycler. The thermal cycler heats and cools the reaction. Okay, a device that is used to heat and cool the reaction. So what are the steps? Steps are the same. First of all, the denaturation that is occur at the 94 degrees centigrade or the 95 degrees centigrade. Okay. Then annealing, it is actually uh, vary between like 48 to 62 or the 65. It depends on the primer. And extension next mainly extension occurs at 72. And these steps are repeated for 30 to 40 cycles on an automated cycler. They can heat and cool the reaction mixture in a short time. So these are the steps. First of all, the then annealing, in which the forward and reverse primers attach with it, and at the end extension that is occurs by TNT. Okay, that's empty. So here is the animation. First of all, the animation, then the primer is attached, annealing occurs, and then after it, extension occurs. Okay. So now we are going towards the one primer designing. Okay, so there are different rules to design the primers. Okay, so we're going through the primer designing rules. First of all, the primer length. It is actually 18 to 25 base pair ideally. Okay, and then the GC content, that is 40 to 60%, and the GC clamps at 3 end is useful, 1 to 3 GC pairs. Okay, what is actually the GC clamp? Uh, our primer have different nucleotides. Okay, A, okay, T, A, like that. Okay, at end there must be the like this type of uh, nucleotides at the end of the primer. Why? Because by this the primer will not be detached. Okay, primer will not be detached when extension occurs. Okay, so that is why at three end, okay, the 5 and that is 3 at three end the GC clamp must be present. That is very useful because it doesn't detach the primer from the DNA template. And the melting temperature. Okay, first of all, the primer length. Why the primer length is very useful here? 18 to 25. Okay, if we decrease the primer length, then non-specificity will be occurred. Okay. Why non-specificity? Because if the number of nucleotides decrease, then the chances of randomly attack with the DNA strand will increase. Okay, but if we increase the uh, nucleotide size, if we increase the nucleotide size, then there is chances we get no bands. Okay, why? Because the chances to attach will decrease, and the melting temperature. Ideal temperature will be 55 to 65 degrees centigrade. Okay. Uh, what is melting temperature? Melting temperature is the temperature where 50% 50% of the DNA of the double-stranded DNA becoming is becoming single strand. Okay. Uh, at melting temperature. Okay. So the difference between the forward and reverse primer must be less than 3 degrees centigrade. Okay. Some literatures have less than 5 degrees centigrade, but ideally it must be less than 3 degrees centigrade. So how we estimate the TM? TM, the simple formula is 2 degrees centigrade is multiplied with the A's present in your primer and the T is present in your primers. Okay, sum it and multiply with the 2, and also the G present in your primer and the C is primary in your C nucleotide primary in your present in your primers. Sum of it and then multiply with 4. Okay, and then at the end make sum of it, and the result we get will be 55 to 65. So the fourth rule is avoid. What should we avoid? The secondary structures, okay, the hairpins, like that type of things occur in your thymus. There must be uh, avoid. Why? Because they make the primers vulnerable. Okay, you get the primer dimers in your gels. Okay, like that. This is the gel. Okay, if you attend my last and the previous workshop, I told about the gel electrophoresis and all the steps of the molecular biology. Okay, in which I told about the primer dimers. Actually, we have like this is the letter. Okay, and actual result must be here. Okay, but we got also this type of bands. This is actually the primer dimers. Why? Because the primers are attached with each other. Okay, not with the strand. Okay, so this type of problems cause the primer dimers. So how it occurs due to secondary structures. Okay, the primer dimers, self dimers, cross dimers, this type of dimers, the your primers will be vulnerable. Long homopolymers like AAAs, if AAAs are present, or like the TTs are present in your in your primer nucleotide, then the chances to attach with each other increase. Okay? Like this. So what they can do, like they can do this is attach with T, this, they attach with, so this will be occur. So they must be avoided. And the complementary at the three ends danger. They also add danger. Like they also cause the damage. The amplicon size must be 100 to 400 degrees 400 base pair for standard PCR and 70 to 150 base pair for qPCR. It is the ideal situation and up to 3 kb for long PCR. And now we talk about the steps of primer design. Okay. Just note this on your notebooks, but I also share you the slides with you in WhatsApp group. Okay. First of all, the step one is get the target sequence. First of all, get the target sequence. Okay, where your target DNA is present. Okay, you get it from the NCBI gene. Okay, there's also you get from the NCBI height and you get from the N sample. These are actually the databases of the DNA where you get your targeted uh, sequence. Okay, and you must have in it fasting format. And the step two is select the target region. Okay, first of all, get the DNA sequence or the gene sequence, and then select the target region. You choose from the exons, you get choose the gene region, you choose the mutation hotspot regions, or you can also choose the full ORF, the depending on experiment. Okay, you can get the full open reading frame, and the most golden point is here is the high specificity. The specificity must be high. Okay, they actually always join to the target region. Okay, there must not be primers that make the non-specific. And the third thing is step three is step three is use an online tools. Okay, in step three, you can use different types of tools. We will talk about in later also, but the main famous tools or the mostly used tools are NCBI Primer-BLAST. Okay, NCBI Primer-BLAST. The second one is Primer 3. We mostly use this one. We also use and the SNAP gene, and at the end IDT oligoanalyzer. Step 4, set the parameters. Okay, set you can set the parameters by the rules we have taught in the previous slides. First of all, inside the primer blast or the primer 3, the product size must be 100 to 400 base pair is the standard PCR, and 70 to 150 if it is the primer size must be 18 to 25. You can select on the in the softwares. We will talk in the next slides. And the TM must be 55 to 65, and the TC content must be 40 to 60. Specificity, choose species, Homo sapiens or the other species according to your interest or your research. And the step 5 is analyze the primers. Get using oligoanalyzer. Okay, after you get the primers, you can analyze it by checking the hairpins. Hairpins must be less. Okay, it must be around 3, and the dimers must be around 5, and the complementarity must be not must be available. Okay, there will be no complementarity. And the step 6 is check the specificity. Okay, blast both primers. Okay, you can blast the both primers to ensure that they bind only to one location in the genome. Okay, they must be joined only into the one location. Okay, must be joined onto the one location. No off-target application. What we check here? The non-specificity. Okay, the non-specificity must be. And at step 7, final primer selection. Select the primer with ideal GC. Okay, that must be 40 to 60, and the TM must be closely to each other. Okay, the difference must be 3, and the ideal primers must be have TM 58 to 60 degrees centigrade, and the self dimers must be low or must be 0 or must be less than 3. And now we have a real life example. First of all, we are going to make or design the pair of primers for sequence like the NM203378. Okay, that in NCBI Genbank. Okay, so that the coding sequence of human myoglobin, it is for human myoglobin. Okay, your research or your genes must be will be amplified using the PCR. Okay, this is actually the scenario. You get the primers from this sequence. The sequence is between 156 to 620. Okay, you select this gene target. You get the gene target. Okay, first step is here, and after getting this, first and second step is you want to get the target region. Then you select the software like the primer 3. Okay, like the primer 3. You pick the primers, you copy 156 to 620. Okay, you just copy it and paste it here. Okay, paste it here and select these ones. Okay, check these ones, pick left primer or the right primer. Don't select this. These are used for molecular diagnostics steps. Okay, don't use it, don't check it here. And then at the fourth step you should check the parameters like the product size must be 464 to 800 like it is like 300. and just not changing it. The primer size must be 18 to 25 or the 27. Optima must be 20. Primer TM must be 55 to 65. Okay. And the product TM don't change. So don't. And the primer GC 20 to 80 and must be optimum like the 40. And self-complementarity must be less than 5. And after it, just click on pick primers. Okay? Just click on pick primers. You get this result like that. You got left primer. Left primer means the forward primer, and the right primer, that is reverse primer. That is start from 3 millimeters and that is start from 77. The length is same. TM is 1659 is the best, and the difference is less than 1. It's actually the less than 1. And the GC content is 50%, and the second one is the 45%. The self-complementarity is 0 of reverse primer, and that is 3. Okay, so this is the best primer you get. If you don't like this, you can scroll down to the bottom. You got different uh, pair of primers. Okay, you can select from it also. After this, you can check it from UCSC. Okay, from UCSC, you can do you can do in silico. You can do in silico PCR and check where your actually primers attached. And then the different resources that are used for general purpose of the PCR primer design. Okay, first of all, the primer 3, we go through it, and the primer 3 plus actually used for the allele specific PCR or the arms PCR. Okay, that are used for those. And the primer Z is used and for the primer and the vector anti-1 advantage. Okay, for real-time PCR, we use NCBI probe database, RT primer database, primer bank, Q primer depot, PCRQ PPD, and the third primer. Okay, now if you have any question, you can ask me in the chat box. Okay, the after it we talk about the PCR optimization. First of all, if you have any question about PCR or the primer designing, you can ask me in the chat box. You have 5 minutes. If you have any question about PCR or the primer designing, you can ask me in the chat box. So let's start. So first of all, PCR. Okay, Myra. Yes, yes. The recording will be available. We will share the recording into the WhatsApp group. Okay. And another, uh, the literature material like the lecture material will also be shared in the WhatsApp group. Okay. If you have also ask me in the WhatsApp group, okay? I am also added in the WhatsApp group. If you have any question, you can ask me in the WhatsApp. Okay, which software is best for designing of allele specific PCR or arm specific? Okay, mostly we use Primer 3 Plus. Okay, Primer 3 Plus is mostly used for designing the allele specific PCR or the arms PCR. Because in allele specific PCR or in the arms PCR, uh, we need in allele specific PCR we need four primers. Okay, the two primers that is actually attached with your target region, and another two region, sorry, the primers are used for to attach first one with the one type of the mutant, or and the second one is uh, with the normal one. Okay, so these primers are actually designed by using primer 3 plus. And in the arms PCR, you know we have to make the restriction enzyme that is actually attached with the target region. Okay, can we need to clean the sequence while copy pasting in primer software? Yes, if you want. Okay, it depends on what you actually want. You want to amplify the whole gene. That is very difficult. But if you want just to amplify two to three exons, okay, or the whole operating frame, then you just copy that sequence, okay. Like you need to sequence of one exons, like the exon 15. Then you just copy the four above and the four uh, bottom lines, okay, and then paste it into the software tool, and then set the parameters, and then click on the pick primers. Okay, so yes, in primer software, when you copy pasting from the NCBI or the ensemble or the different types of software that DNA databases, you need to clean the sequence. But in that uh, manner, if you want to clean the sequence in between, that is not important, and it is not recommended to you. But yes, to select the specific target region is very important, because by this the primers will be designed into your target region, okay, not from the whole DNA sequence. Also, you get the answer. If you don't get the answer, you can ask me again. I will explain you briefly again. Okay, welcome. If you have any question, you can ask me in the chat box. If another participants have question, you can ask me in the chat box. Okay, now let's go towards the next part of the session of the master class. Now we talk about the PCR optimization, and it is definitely very important. Okay, and we talk about the different steps or the you can say the rules or in the optimization and where you have to focus. So kindly note in your notebooks or check it in the recordings again and again. And if you don't get it, you can ask me the questions in the WhatsApp group, or you can also ask me in the personal chat. I will definitely answer. And if you want to, if you want that I recommend or I suggest you something, get the best results from the PCRs you are doing. And if you get no results, I will definitely help you out in that manner. Uh, Now I am going to share you the screen to you. So we are going to do the second part of the our session. Also, you see the screen. If you don't see the screen, kindly message that. Okay, now the last part module of our workshop is PCR optimization. Okay, PCR optimization. So, what are actually the common problems we face? Okay, we face like we get no bands. Okay? In health, we get no bands, or like the faint bands, we get the very dim bands, or we get the extra bands, like we get hematite, or we get non-specific. Okay, or like we get smearing or the non-specific bands. Okay, these are actually the common problems we face. So to go, to rule out or to troubleshoot these problems, we do primer optimization. Okay, the primer optimization or the PCR optimization. Okay, so what are the optimization steps? Now we are going towards the optimization steps. First step of PCR optimization is the annealing temperature optimization. Okay, the main focus on the PCR optimization is annealing temperature. Okay, this one is also called primer optimization. Okay, a primer optimization in which we use graded PCR. Okay, mostly we use graded PCR. If we get no results, then we go towards the touchdown PCR. Okay, the touchdown. If we get no results from it, and then we go towards the touchdown PCR. So, what are the rules for the annealing temperature optimization is first of all gradient range. The gradient range must be plus minus 3, okay, from the primer Tn. Like the primer Tn is 58, then the minus 3 and the plus 3 of your Tn must be like 5, and the plus 3 is 61, okay. So the range must be 55 to 61. So for example, the Tm is 58 and the run gradient must be 55 to 61. If you get the results, you can also, if you have a time, you can also do the plus minus 2. Okay, it makes more stringent results and you can select from it more precisely. Choose temperature where you get the clear signal band. Okay, you get strong intensity and you get no non-specific band. Okay, select that temperature. Okay, the second step of the optimization is MgCl₂, magnesium chloride optimization. Magnesium affects the polymerase activity. Okay, the magnesium present in your PCR master mix actually affects your polymerase activity. If very low Mg for the magnesium ion, then we get weak or no bands. Okay, and if it is high magnesium ions, then you get non-specific bands or like you get the smears. So first of all, start from 1.5 millimolar, then go towards the 2 millimolar, and then towards the 2.5. So this is the ideal situation. You may get the best result at 1.5, or you get to at the 2.0, or you get the 2.5. Okay, template DNA optimization. The third step of optimization is template DNA optimization. If you get the if you use the too much DNA, okay, then you get the non-specific binding. Okay, just note on your notebooks. If you have use too much DNA, then you get the non-specific binding. And if it is too little DNA, then you get the faint band. Typically, 50 to 100 nanogram genomic DNA and 1 to 10 nanogram plasmid DNA is mostly ideally used. Okay, the primer concentration. Use 0.1 micromolar to 0.5 micromolar. Okay, what is actually the sorry, the picomolar? We mostly use 2 picomolar. Why? Because 2 picomoles are enough for 30 to 40 cycles. What happened? Okay, if the concentration of the primer is very high, it makes primer dimers. Okay, and if the concentration is very low, it makes the weak band. So must be in between. You get the best bands intensity and you get no primer dimer. And in the sixth, that's the sixth one, DNTP optimization. The standard DNTP is 200 micromolar or the picomolar. Okay, the DNTP or the 200 DNTP are enough for 30 to 40 cycles. Okay, and enough for extend your target region. If it is high, it increase the error rate. The error rate must be increased, and it we also get non-specific binding. And at the and polymerase choice. The polymerase must be we use mostly we use in the routine we use tag, okay, because they can bear the denaturation step. Okay, they doesn't degrade at the DNA. And if we are going towards the cloning, then we choose high fidelity polymerase. Okay, high fidelity polymerase for cloning, like the PFUQ5. And the cycle numbers, we chose 25 to 30 cycles. If we chose 15 to 20, then it's called short PCR. And the mostly we use 25 to 35 cycles. If more cycles, we get non-specific formula. And the gel running conditions, okay, must be must be in your mind. Okay, we use 2% agarose if your amplicon size is less than 500. Okay, must be in your mind. If it is greater than 500 base pairs, then choose 1%. And the voltage must be 80 to 110 volt. And the time must be 45 to 60 minutes. If we increase voltage and also increase time, then what will be happen? By this, okay, now we are going towards the real time example. Okay. Okay, first of all, the time is just 10 minutes left. Okay, then the call will be end. Okay, now I am going to share you the news link with. Okay, in the WhatsApp group, I am going to share you the new link. Kindly join that. Leave this meeting and join the new link I am going to share you in the WhatsApp group. Kindly join the new link I am going to share you on the. Okay, now I'm going to share you the screen for you. So by this we're going to. Okay, first of all, type NCBI. because the speed of the internet is very slow, so kindly bear from it. Okay, actually the coming from Raja, the speed is very slow. So if you want to design the primers from me, I will definitely design the primers for you. You can just ask me in the personal chats. I will definitely help you out. So if this will work, I will show you the real examples how to design the primer. Okay, okay. If NCBI is not open, I will use the ones from my. So this is actually the sequence. Okay, you get from NCBI. Okay, it is just the first step, first step of it. Okay, so like we want to design the primers from axon. Okay, so this is the first step format. Okay, and these one are actually the axon. This red one is actually the axon 14, and this one is the axon 15. Okay, and I'm going to make the primers from it. Okay, so I just copy the above and the lower of the sequence from your target sequence. So after it, just paste it in the in primer 3. Okay, so just check this one. Okay, check this, not the uh, middle one. And the primer size 18 to 23. Okay, or what? The primer TM is 57 to 2. Okay. That is good. And the product TM and the primer GC content must be 30 to 70 and the primer 50. Okay, it doesn't change in okay. If you want, you can change in it, and but the software is just the artificially designed to make it good primers. Okay, so after it, just click and click on the pick primer. Okay, you get the output, and the left primer is GCACTGC7. Okay, and the right primer is this one. It is start from this and it start from this and length is 20. Okay, good one. TM is 58.96 and 58.92. The difference is less than y. Basic content is 50% and the 50%. Okay, good one. Uh, okay, and the self dimers is 0. 3 prime complementarity is 0. Hairpins are 0. Okay, the best primers we get. If we don't like it, you can also select from these ones. Okay, but these primers are best. Okay, now you just write here calc, oligo calc. Okay, and then select University of California, Berkeley oligo calc. Okay, is open and just copy the left primer and paste it here. Okay, and click on calculate. You just verify it from LicoCAD. Length is 20, melting temperature is 52. Here we get the melting temperature 50. Okay, so there is a difference. So this is the actual calculator where you can calculate. The second data is 50. Okay, it's good. And OD1 is for joy, let's see. Okay, and just copy the right primer and then calculate. Okay, we get length 20, melting pressure 52, basic content 50, and molecular weight 6152, and OD of 1 is equal to 4537. And the melting pressure is 52. Okay, just or get the screenshot into and save it in the folders. Okay, then you can also go towards the blast, but it is very difficult to open it because the speed of the internet is over here. But I'm going to try on my side. Not open. We will do this copy. Going to open the CSC, but first of all, it must be open. Okay, if it is not open, actually what we do in Blast is we check that primers we designed that primers were designed is actually bind or actually uh, designed for the specific target region or not. Okay, we do the blast for that. Okay, and the blast is not open, but UCSC is okay. Then select add in silico. Okay, select in silico. Click on it. Okay, select the species like the human one. Genome summary. Okay, target genome summary of the gene code gene and paste the forward primer and then copy the reverse primer and paste it in reverse primer. Okay, and then click on it, submit. Okay, first of all, open it. I'm going to show you that the gene in which the primers must be attached, it must be at chromosome 5, okay, in the Homo sapiens, okay, and in the exon 15 or around the exon 15. So we get the in silico PCR result. Just click on it. Okay, coming from Raja, last is not opened yet. I'm going to open it, but it's not opened. If it is open, I will. Okay, okay, open. Okay, I will show you the steps here. But first of all, I'm going to show you the in silico PCR results. Okay, you can check the primers is actually attached at the chromosome 5. Okay, you can see here it is attached at chromosome 5. Okay, and at the Q arm, the long arm at the at the 2.82, like at the same band where we actually want to get the results. Actually attach here at that red point. So the primers we designed is actually the same we want. So by this we can check the we can validate our primers. Okay, in blast we actually do blast to check that the primers we design or we select is actually bind at the specific point of your target region. Okay, so how you can do this? You can select the nucleotide blast. Okay, this one. Okay, it must be open. Okay. Now I'm going to paste the forward primer here. Okay. Now click on it on the blast. But the internet service is very poor and I think it is very difficult to get it done because the internet is not any so but okay. When you get the primers, okay, then you just check that these primers are attached at the same point or the same region where actual you are focusing, okay, or your target region. The primers must be attached at the target region. How you can check it, how you can validate it, how you can analyze it by using UCSC. How you can do it? First of all, type UCSC on the Google. Okay, then click on UCSC genome browser. You get these one results. Okay, just click on in silico PCR. In silico PCR. This web page will be open. Just paste forward primer, copy, paste here, and then copy UCS left primer and paste in reverse primer, and then click on submit. Okay, so these primers must be present in chromosome 5 and at the long arm 22.2. So like this red one is actually the primer attachment point. So it is attached at the chromosome 5 Q22.2. Okay. By this you can confirm that your primer is attached at the target region. Okay. So we are validating that in the UCSC that our primers is attached at the specific point. Okay. Okay, the blast is not working, so sorry for that. I will share you the slides. You can check it by yourself. I have explained all the concepts with you. So today session is over. Thank you so much for joining it. If you have any question, you can ask me in the chat box. Okay, I am here. If you have any question, you can ask me in the chat box. So this is the question and answer section. If you have any question, you can ask. I will I will definitely give you the answer of it. If you have any personal questions, like if you have, you are stuck in your PCR problems, or like the PCR optimization or the primer designing for the research, or for the uh, or in the lab, you can ask me in the WhatsApp group, or you can ask me by getting the my phone number or the WhatsApp number from the WhatsApp group, and you can ask me in the personal chats. I will definitely, I will delighted to help you out. And if you get the solution from me, I will definitely help you from it. Now the session is over. If you have any question, you can ask me in the chat box, or you can ask me by unmute the chat. Okay, Bashir Ahmed Shaheen, if you have any question, you can ask me. I think so. I think so. No one has a question. Am I right? If you have any question, you can ask me in the chat box. If you have question after this session or the or this workshop, you can ask me in the WhatsApp group also. Okay. Okay, Bashir Ahmed Shaheen has question. How can newly synthesized primers will be developed? Okay. First of all, when you save the primers, you just dilute it by adding 200 microliter in the separate tube. Okay, 200 microliter water in the forward primer and 200 microliter uh, uh, nucleus water in reverse primer. Okay, you make you just lyophilize the primers. After it, you use two apple drops. Okay, first apple drop for forward primer, second apple drop the new apple drop, and the second apple drop for new apple drop for reverse primer. Okay, and you add 10 microliter of the forward primer in the forward primer apple drop, and 10 microliter of the reverse primer in the reverse primer apple drop, and add 90 microliter of water, nucleus free water. Okay, you ask which water should be used. The nucleus free water, nucleus free water, by which the nucleus will not be, the DNA will not be degraded, and you add 90 microliter in the forward primer and 90 microliter water, nucleus free water into the reverse primer. Okay, and by this are making you are making the 10 picomole of the primers. Okay, you are diluting the primers to make the 10 picomole of the primers. Okay, and then from it you use to 0.2 to 0.3 microliter for the PCR. Okay, and by this you used just 2 picomole, and 2 picomole is enough for 40 cycles. Okay, you got the answer, Shahan? Kindly ask me again how many times the final diluted primer used the freeze. Uh, when you make the dilution, okay, you can use it directly. Okay, but if you want to freeze it, you can freeze it at -20. Okay, and you can use it for longer 30 to 40 minutes for sorry 30 to 40 days. If you want to freeze if you want to store it like like 5 to 6 months, you can store it in minus 40 degrees. Okay, so you get the answer. Okay, welcome, welcome. If you have any question, if another person have any question, you can ask me in the chat box. Yes, the recordings will be shared to you tomorrow. I will definitely share the recordings to you in the WhatsApp group. Yes, yes, I have experience about the arms PCR, in which we make tetra primers for primers, okay, for SNP detection. I, yes, I have experience. I have done primer designing for arms PCR for my participant, one participant that have uh, asked me to make the primers for them. Okay, I provide the services for in which I help others to get the research to done the research time. Okay, so by this I design the primers for them, and also I do the PCR optimization for them. If you want me to design the primer, you can ask me in the personal chats. I will definitely help you out. Okay, if you have any question, you can ask me in the chat box. And if you have not any question, so this is the end of the session. Thank you so much. You can, if you have no any question, you can leave the session. Thank you so much for being the part of this journey. Okay, thank you so much. This as time for the session is now over. Now you can leave the session. Okay, thank you so much. If you now I'm repeat this. If you have any question about the primer designing and the PCR optimization, or about the molecular biology techniques, or about the research, you can ask me in the WhatsApp group or in the personal chats. Okay. Thank you so much.